Have you ever wondered why some people carry invisible scars that never seem to heal? Why do certain childhoods create wounds that echo through decades, shaping every decision, every relationship, every moment of despair? Scientists just discovered the hidden chemical your brain creates when trauma strikes, and it's been driving you toward darkness ever since. When you were 5 years old and your father's voice shattered the walls, your brain didn't just record the memory, it began producing something far more sinister. When you were 7 and learned that love comes with conditions, with fear, with silence, your neurons didn't simply store the experience. They started manufacturing a chemical weapon against your future self. And when you were 9, sitting alone in a room that should have been safe, your hippocampus, the memory center of your brain, began building the architecture of your adult depression. This isn't poetic language. This is neuroscience. For decades, we've understood that childhood trauma predicts adult depression. The statistics are horrifying. 60% of American adults diagnosed with major depression experienced some form of adversity before they turned 18. Two-thirds of people who attempt suicide carry memories of childhood neglect, abuse, or abandonment. We've known the correlation for years, but correlation isn't causation. We never understood the mechanism. We never saw the invisible thread connecting a terrified child to a suicidal adult 30 years later. Until November 2025, when neuroscientists at Columbia University and McGill University discovered the missing link, a brain chemical called SGK1. SGK1, serum and glucocorticoid regulated kinase 1, is a stress response protein. Your body produces it when cortisol floods your system, when your fight or flight mechanism activates, when your nervous system interprets the world as dangerous. In healthy brains, SGK1 levels rise temporarily during stress and then return to baseline. It's part of your survival machinery, an evolutionary gift that helped your ancestors escape predators and survive hostile environments. But in the brains of people who experience childhood trauma, something fundamentally breaks. The SGK1 factory never shuts down. The research team, led by Dr. Christoph Anaker, examined the brain tissue of adults who died by suicide. What they found was extraordinary and devastating in equal measure. SGK1 levels were significantly elevated in every suicide victim they studied. But the highest concentrations, up to twice the levels found in other suicide cases, appeared in the brains of people who had documented histories of childhood adversity. Think about that for a moment. A chemical fingerprint of childhood trauma, still present in the adult brain decades later. Still active, still destroying. Your 8-year-old self, terrified and alone, reached forward through time and changed the molecular structure of your 40-year-old brain. The researchers didn't stop there. They analyzed genetic data from children currently experiencing adversity, tracking them through their teenage years. They discovered that certain genetic variants increase SGK1 production in response to stress. And children carrying these variants, children whose bodies naturally produce more SGK1 when traumatized, showed significantly higher rates of depression by their teenage years. The chemical doesn't just correlate with depression, it predicts it, it causes it. This explains something that has puzzled psychiatrists for generations. Why do standard antidepressants often fail people with childhood trauma? SSRI antidepressants, selective serotonin reuptake inhibitors, work by increasing serotonin availability in the brain. For many people with depression, this approach is effective. The fog lifts, the weight decreases, life becomes livable again. But for people with histories of childhood abuse or neglect, SSRIs frequently provide minimal relief or none at all. Psychiatrists have observed this pattern for decades without understanding why. patients would sit in offices describing the same symptoms as other depressed individuals, receiving the same medications, and experiencing vastly different outcomes. The lucky ones found modest improvement. The unlucky ones found nothing, or worse, discovered that hope itself can become a form of torture when treatment after treatment fails. The answer is SGK1. The biological pathway from trauma to depression doesn't run primarily through serotonin. It runs through stress response proteins that SSRIs don't touch. You're treating the wrong mechanism. You're trying to fix a broken window when the foundation is crumbling. It's like giving aspirin to someone with a broken leg. You might reduce some inflammation, but you're not addressing the structural damage. Dr. Aniker's team had suspected SGK1's involvement 10 years ago when they found elevated levels in the blood of unmedicated patients with depression. But blood chemistry isn't brain chemistry. The breakthrough came when they gained access to postmortem brain tissue and could measure SGK1 directly in the hippocampus, the brain region responsible for memory formation, emotional regulation, and stress response. The hippocampus is where your childhood lives. Every traumatic memory, every moment of fear, every instance where safety should have existed but didn't, these experiences are encoded in hippocampal neurons. The scent of alcohol on breath, the sound of a door slamming, the feeling of being small and powerless while adult voices raged, the cold understanding that nobody is coming to help. All of it stored in neural networks, reactivating throughout your life in response to triggers you may not even consciously recognize. And in the brains of trauma survivors, those neurons are bathed in excessive SGK1, like plants drowning in toxic water. What does SGK1 actually do at the molecular level? That interferes with neurogenesis, the birth of new neurons. Your hippocampus is one of the few brain regions that continues producing new cells throughout your life. These new neurons are essential for emotional flexibility, for recovering from stress, for adapting to new circumstances. They represent your brain's capacity for renewal, for learning that the past doesn't have to determine the future. But elevated SGK1 suppresses neurogenesis. It prevents your brain from healing itself. It's as if trauma not only wounds you, but also prevents the wound from closing. This creates a vicious cycle that operates beneath conscious awareness. Childhood trauma increases SGK1. Elevated SGK1 reduces neurogenesis. Fewer new neurons mean decreased ability to process and recover from stress, which triggers more SGK1 production, which further reduces neurogenesis. The brain becomes trapped in a self-reinforcing pattern of vulnerability, like a physiological echo chamber where past suffering continuously amplifies present pain. To test their hypothesis, the researchers conducted experiments with mice. They subjected the animals to chronic stress, a laboratory equivalent of sustained adversity. The protocol involved unpredictable stressors, social defeat, environmental instability, the rodent version of growing up in chaos. As predicted, the mice developed behaviors analogous to human depression: social withdrawal, anhedonia, loss of pleasure in previously enjoyed activities, learned helplessness. But when the researchers administered SGK1 inhibitors, drugs that block the protein's activity, something remarkable happened. Even under continued stress, the mice didn't develop depressive behaviors. The SGK1 inhibitor broke the cycle. It was like installing a circuit breaker in an overloaded electrical system. This suggests an extraordinary possibility: trauma-specific antidepressants. Currently, pharmaceutical companies are already developing SGK1 inhibitors for other medical conditions, including atrial fibrillation and certain cancers. The medications exist. They're being tested in human trials for cardiac applications. They could potentially be repurposed for depression treatment within years, not decades. For the first time, We might have pharmacological interventions designed specifically for people whose depression originates from childhood adversity, rather than forcing everyone through the same trial and error process with medications designed for different biological mechanisms. But there's another layer to this discovery: genetic screening. Remember those genetic variants that increase SGK1 production? The researchers proposed that individuals seeking depression treatment could undergo genetic testing to determine whether they carry SGK1 increasing alleles. If they do, they would be prime candidates for SGK1 targeted medications. This is precision psychiatry, matching specific biological mechanisms to specific treatments, the way oncologists increasingly match cancer treatments to tumor genetics. It represents a fundamental shift from treating symptoms to treating causes. The implications extend far beyond individual treatment. Understanding the SGK1 pathway illuminates the broader relationship between early adversity and lifelong vulnerability. It explains why some people develop resilience after childhood hardship, while others struggle with persistent mental health challenges. Resilience isn't just psychological fortitude or positive thinking, it's neurochemical. Some people's brains naturally produce less SGK1 in response to stress, either through genetic factors or through protective environmental influences. Others produce more. The difference can determine whether childhood trauma becomes a permanent sentence or a survivable chapter. This also reframes how we think about prevention. If we can identify children with genetic predispositions toward high SGK1 production, and if we can intervene early, either through medication, therapeutic interventions, or environmental modifications that reduce stress exposure, we might prevent the cascade from trauma to depression to suicidality before it fully develops. We might be able to identify at-risk children and provide targeted support before the neurochemical changes become entrenched. Consider the broader landscape of childhood adversity and its many forms. Physical abuse that teaches a child their body isn't safe. Sexual abuse that corrupts the fundamental understanding of trust and intimacy. Emotional neglect that communicates you are not worth attention or care. Witnessing domestic violence that demonstrates love and violence can coexist. Growing up with parents struggling with addiction or severe mental illness, learning that adult behavior is unpredictable and frightening. Food insecurity that creates chronic stress around basic survival. Housing instability that prevents the development of security and routine. These experiences don't just hurt in the moment. They reprogram the brain's stress response systems. They change gene expression patterns through epigenetic modifications. They alter the fundamental architecture of emotion and cognition. Your nervous system, trying desperately to adapt to a threatening environment, becomes hypersensitive. Every ambiguous social cue gets interpreted as danger. Every disappointment feels catastrophic. Every setback confirms your worst beliefs about yourself and the world. And underneath all of it, SGK1 levels remain elevated, suppressing the neurogenesis that might allow your brain to update its models of reality. And society has largely treated these outcomes as inevitable. We acknowledge that childhood trauma increases depression risk, shrug sympathetically, and prescribe the same medications we use for people without trauma histories. When those medications fail, we label the patient treatment resistant and move on to the next intervention, hoping something works. We pathologize the survivor rather than examining the inadequacy of our treatments. But the SGK1 research reveals that we've been operating with an incomplete map of depression. Depression isn't a single condition, it's a collection of distinct biological pathways that produce similar symptoms. The depression of someone whose serotonin system malfunctions is neurochemically different from the depression of someone whose stress response proteins are chronically elevated. Same symptoms on the surface: the fatigue, the hopelessness, the anhedonia, but different underlying causes, different solutions. This matters enormously for suicide prevention. Suicide isn't typically an impulsive act, despite how it may appear to shocked loved ones. It's the culmination of sustained suffering, of trying every available treatment and finding no relief, of believing that the pain will never end because nothing has ever made it end. Many suicide victims spent years in treatment. They tried multiple medications. They engaged in therapy. They wanted to live. They fought to live. But the tools we gave them couldn't address the underlying mechanism driving their despair. They were fighting with inadequate weapons against an enemy we hadn't properly identified. The SGK1 pathway offers hope that we might finally have tools that work for this specific population. Not for everyone. Depression is too complex for any single intervention to help all patients. But for the substantial population of people whose depression originates from childhood trauma, that population is enormous and has been chronically underserved by existing treatments. There's something almost poetic about this discovery, even in its darkness. The brain keeps receipts. Every trauma leaves a molecular signature, a chemical memory that persists long after the conscious mind has processed, or tried to process, the event. Your body remembers what your mind tries to forget, and in that remembering, it continues to protect you from dangers that no longer exist, flooding your system with stress responses to threats that have passed. SGK1 is your brain's way of saying, "The world hurt you once, and I will never let you be unprepared again." But that hypervigilance comes at a devastating cost. The same mechanism that keeps you alert to danger also prevents you from experiencing safety, from forming secure attachments, from believing that tomorrow might be better than today. The future holds possibility. Clinical trials will take time. Safety testing will take time. Regulatory approval will take time. But the pathway is clear. The mechanism is identified. The target is visible. For people who have spent decades wondering why they can't heal, why they can't escape the gravitational pull of their past, this research offers something precious: explanation. You're not broken. You're not weak. Your brain is operating exactly as it was programmed to operate by circumstances beyond your control. And now, finally, we understand the programming well enough to contemplate rewriting it. If you found this information valuable, subscribe to this channel, hit the like button, and comment your city below. Let's build a community of people who understand that mental health is brain health, and that science, is slowly, steadily illuminating the darkness we've carried for too long.